So, maligayang araw everyone. So, we are here to give you a video presentation about gel electrophoresis. But before that, let me introduce to you to what gel electrophoresis is. So, this is a technique used to separate DNA fragments according to their size and charges. So, in our presentation, we will show you a very simple experiment using dyes, batteries. So, please watch our video and I hope you will understand what electrophoresis is. Here are the materials and equipments needed for this experiment. Red and black wires. 9 volts batteries, gel chamber with agarose, dye and micropipette tips. Micropipette and carbon paper. Falcon tube and TBE solution. Use the falcon tube to fill the gel chamber with TBE solution. Fill the gel chamber such that the TBE level is above the divider separating the agarose rather, which means that the TBE have to submerge and overflow from the agarose. Place the carbon paper at both ends of the gel chamber. Ensure that the carbon paper is submerged in the TBE solution. Clip the carbon paper with wire. Repeat and do the same for the other end of the chamber. So you will just repeat the process with the other side of the chamber. Attach 5 9 volts batteries together. Ensure that the negative is connected with the positive. So we'll just connect the batteries from negative to positive. Attach the micropipette tip and adjust volume to 4 microliter. Extract the dye. Remember to first stop, dip the tip, second stop, then full release. Place each dye into the chamber. Ensure that the tip is not piercing through the agarose. Repeat the whole pipetting process for the different dyes. Remember to use different micropipette tip for the different dyes. So you should use different pipette tips for different dyes so that the color will not interchange with each other. So, and it's also clean. Last dye na. Most of the dye are negatively charged. Therefore, ensure that the wire closest to the dye is negatively charged. It is negative to positive. Connect the wire to batteries and the current will begin to flow. When the battery is running, small bubbles will begin to be emitted at the negative side of the gel chamber. This is because hydrogen gas is being released. The result after 5 minutes, result after 10 minutes, after 15 minutes, after 20 minutes, after 25 minutes, result after 30 minutes, extent of the gap may differ. And now we will show you a video presentation using a hemacytometer to count cells.
Cells. It's a modified microscope slide containing two identical wells or chambers into which a small volume of cell suspension is pipetted. We have already removed 100 microliters of our cell suspension and placed it in a microcentrifuge tube. Dilute the suspension by adding 100 microliters of trypan blue. Trypan blue is a dye that helps us distinguish between living and dead cells. The dye passes through the membranes of dead cells, so they will appear blue under a microscope. Living cells exclude and will appear mostly clear. Load both chambers by pipetting the suspension under the cover slip. Now place the hemocytometer under the microscope. Each chamber is divided into a grid pattern consisting of 9 large squares. Each square has the same dimensions and contains 10 to the negative 4th power milliliters of suspension. The rules for counting cells sometimes differ from lab to lab. In our lab, we count cells in the 4 large corner squares and the center square. Let's count the cells in the first square. 1, 2, 3, 4, 5, 6, 7, 8. So what about the cells that are touching the outside boundaries of the square? In our lab, we count the cells that touch the top and left boundaries, and we ignore the cells that touch the right and bottom boundaries. 9, 10. We need to count the number of both living and dead cells. Remember, the dead cells will appear blue. Occasionally, you will see artifacts, objects or debris that appear blurry and don't have a well-defined shape. This is an example of an artifact. We won't include it in our count. Proper storage, cleaning, and handling of the hemocytometer will minimize the number of artifacts. There are 10 viable cells and 1 non-viable cell in the first square. Now, the top right square. There are 9 viable cells and no non-viable cells. Next, let's count the bottom right square. There are 11 viable cells and no non-viable cells. And now the bottom left square. There are 10 viable cells and 2 non-viable cells. And finally, let's count the cells in the center square. Sometimes cells will appear as clumps or small groups. It may be difficult to determine exactly how many cells are in a group. The method of counting clumps of cells differs from lab to lab, so be sure to follow the procedure in your lab. We will count this clump as 2 cells. There are 14 viable cells and no non-viable cells in the center square. The total number of viable or living cells from all 5 squares is 54. The total number of non-viable cells is 3. Now that we have counted our cells, there are several calculations we need to perform. First, let's calculate the percentage of viable cells. Here's the formula. 54 viable cells divided by 57 the total number of cells, gives us 0.947. Multiply by 100 and the percentage of viable cells is 94.7%. Next, let's determine the average number of cells per square. We counted 54 viable cells. We divide 54 by 5 because we counted in 5 squares. The average number of cells per square is 10.8 cells. Now let's calculate the dilution factor. The dilution equals the final volume divided by the volume of cells. Our final volume is 200 microliters because we started with 100 microliters of cells and added another 100 microliters of trypan blue. 200 divided by 100 is 2. Therefore, the dilution factor is 2. Next, we need to calculate the concentration of viable cells, the number of living cells per milliliter. Our average count per square is 10.8. The dilution factor is 2. 10.8 times 2 times 10 to the fourth power equals 216,000 cells per milliliter. We can write the concentration using scientific notation as 2.16 times 10 to the fifth power cells per milliliter. From our calculations, we now know the concentration of cells in our culture is 216,000 cells per milliliter, and approximately 94.7% are viable.
Thank you for watching, guys. We would like to give thanks to Carolina Education Consultancy and Bio Network for the wonderful videos. Thank you again. God bless.